Gastrointestinal Physiology, Liver Physiology. Welcome to the Everything Surgery podcast, where today we delve into the fascinating world of gastrointestinal physiology, focusing on the liver. Often described as the metabolic powerhouse of the body, the liver serves as a central hub, intricately connected to both the digestive system and the systemic circulation, and its physiological functions are as diverse as they are essential. These range from metabolism and detoxification to protein synthesis and digestion. The liver, the body's metabolic central processing unit. The liver should not be considered solely a digestive organ. Rather, it plays a pivotal role in maintaining the body's overall metabolic balance. Its unique anatomy, centered around the hepatic lobule, underpins its immense functional capacity. Functional anatomy of the liver. The structural unit of the liver can be understood through several models. The classic lobule is a hexagonal structure centered on a central vein, which drains towards the hepatic vein. At each corner of this hexagon, you will find a portal triad consisting of a portal venule, a hepatic arteriole, and a bile ductule. Blood from the portal venule, which is rich in nutrients, and blood from the hepatic arteriole, which is high in oxygen, combine and flow through sinusoids toward the central vein. As blood passes through, hepatocytes, the functional cells of the liver, process its contents. Another model, the portal lobule, emphasizes the liver's exocrine function in bile secretion. This structure is a triangular area centered on a portal triad, with central veins at its corners, delineating the territory drained by a single bile duct. Perhaps the most functionally relevant model is the liver acinus. This is an oval region oriented around a line connecting two portal triads and is divided into three zones based on the levels of oxygen and nutrients. Zone 1, closest to the blood supply, is highly active in oxidative metabolism, fatty acid oxidation, and the production of new glucose. Zone 2 serves as a transitional area, while Zone 3, furthest from the blood supply, is the site of glycolysis, fat synthesis, and detoxification processes, making it especially vulnerable to injury from lack of oxygen or toxins. The hepatocyte. The multifunctional cell hepatocytes are the primary cells responsible for the liver's vast array of tasks. Their structure is polarized, with the sinusoidal membrane facing blood-filled sinusoids to facilitate exchange with the blood, and the canalicular membrane facing the bile canaliculi to enable secretion of bile components. Key physiological functions of the liver. Carbohydrate metabolism. The liver acts as the body's main regulator of blood glucose. During fasting, it breaks down stored glycogen to release glucose. After a meal, it stores excess glucose as glycogen. When fasting is prolonged, it synthesizes new glucose from lactate, glycerol, and amino acids. Additionally, the liver converts galactose and fructose into glucose. Protein metabolism. The liver is responsible for synthesizing and releasing most of the plasma proteins, including albumin, which maintains blood pressure and transports substances, and clotting factors such as fibrinogen and prothrombin. It also produces transport proteins for iron and copper. Through deamination and the urea cycle, the liver converts toxic ammonia generated from protein breakdown into urea, allowing for safe excretion by the kidneys, a vital detoxification function. Lipid metabolism. The liver breaks down fatty acids for energy, particularly in areas closest to the blood supply. It also synthesizes new fatty acids and triglycerides from carbohydrates in areas further from the blood supply. During periods of starvation, the liver produces ketone bodies from fatty acids, which serve as an alternative fuel source for the brain and other tissues. The liver is also the main site for cholesterol and lipoprotein synthesis, packaging cholesterol into very low-density lipoproteins for distribution, and producing high-density lipoproteins. It converts cholesterol into bile acids as well. Bile synthesis and secretion. Bile serves both as a digestive secretion and as a means of excreting waste. It contains bile salts, which are synthesized from cholesterol and are crucial for emulsifying dietary fats, enabling their digestion and absorption. Phospholipids support micelle formation. Cholesterol is excreted in bile, and bilirubin, the breakdown product of hemoglobin, is conjugated in the liver to make it water-soluble and serves as the main pigment in bile. Bicarbonate is added to bile by the liver and its ducts to create a solution rich in bicarbonate ions. Through a highly efficient process known as enterohepatic circulation, most bile salts are reabsorbed in the terminal part of the small intestine and returned to the liver for reuse. Detoxification and biotransformation. The liver plays a crucial role in modifying potentially harmful substances, such as drugs, toxins, and hormones, to render them less active and more water-soluble for excretion. This involves two main phases. Phase 1 reactions, primarily handled by the cytochrome P450 system, which involve oxidation, reduction, or hydrolysis, and phase 2 reactions, which involve conjugation with molecules like glucuronate or sulfate, generally inactivating the compound and enhancing its excretion. Conjugation of bilirubin is a classic example of a phase 2 reaction. Storage. The liver acts as a storage depot for essential vitamins such as A, D, and B12, as well as minerals like iron. It also stores glycogen, providing a readily available energy source. Immune function. The liver houses resident macrophages known as Kupffer cells within its sinusoids. These cells are responsible for phagocytosing and removing bacteria, endotoxins, and other debris from the blood arriving from the gut, acting as a vital filter for the body's defense system. Regulation of liver blood flow in the portal system. The liver receives a dual blood supply. The portal vein brings nutrient-rich but partially deoxygenated blood from the gastrointestinal tract, spleen, and pancreas, while the hepatic artery supplies oxygen-rich blood. The liver has an autoregulatory mechanism whereby if blood flow from the portal vein decreases, blood flow from the hepatic artery increases to compensate, maintaining a stable overall flow. Any obstruction of blood flow through the liver, such as in cirrhosis, can result in increased pressure in the portal system, leading to complications such as fluid accumulation in the abdominal cavity or dilated and fragile veins in the esophagus and stomach. Summary: the liver is an integrative center. To summarize, the liver's primary role is in processing nutrients absorbed from the digestive tract. After eating, it stores glucose as glycogen and converts excess glucose to fat, synthesizes plasma proteins and deaminates amino acids, and creates triglycerides and cholesterol for distribution. During fasting, it releases glucose through the breakdown of glycogen and the production of new glucose, utilizes amino acids for energy and produces urea, and oxidizes fatty acids for energy while generating ketone bodies. In essence, the liver serves as the body's ultimate processing plant, seamlessly transitioning between building up and breaking down substances to maintain a constant supply of energy and essential building blocks, detoxifying the blood, and producing crucial proteins and digestive fluids. Thank you for joining us in this deep dive into liver physiology. If you enjoy our content, please like, share, and subscribe. This is the Everything Surgery Podcast, where we dissect the extraordinary.